Hi, this is Menaka. I'm doing my research on cattle genomics at Sastra Deemed University, Tamil Nadu, India. I'm going to present my poster in the title of Integrative Genome-Wide Analysis Identify Immune Signature in Indigenous Cattle, which is Kangayam, Tharparkar, Sahiwal and Karan versus exotic cattle, Holstein, Friesian and crossbred cattle, Karan. So when coming to the introduction part, the Boston cattle native to India offer multiple advantages over Boston cattle. So these Indian cattle, based on their environmental conditions, they evolved through natural selection process and survive under extreme stress and resistant to multiple diseases. Therefore, identifying unique immune signature that can support breeding decision for better immunity. So the genome-wide comparative analysis across these three groups could uncover the immune signature that could be responsible for disease-resistant trait in individual strains. So we did whole genome sequencing of these six traits using Illumina Nova 6000 platform and Redcliffe Labs in Noida. For sequencing, we collected samples from three different locations for each breed, and each location we collected six samples. At the end, we pooled these six into one, and finally we got 18 pooled samples representing these six breeds. So this is the pipeline I followed to identify immune signature in indigenous states. First, I assess the quality of positive files using positivism and remove low quality reads and adapter contamination using chromatic and mapping these reads with two different reference genomes that deposited in ECCB. One is Taurine reference genome, another one is Citizen reference genome with BWM2. And I did sorting and removing the duplicates, and I called the variant using Delhi and Freebase, and I did variant expression and normalization, and I annotated those variant with slip and copy number variant with ensemble. And I did functional enrichment analysis, David, and I did network analysis with string and cytoscape. In addition to that, I did FST statistic test to identify the genetic differentiation across these three groups. So this map representing number of variants which identified by Daily and tree-based. That basically annotate the variant based on four different impacts. One is high, moderate, modifier, and low. High means the particular variant causes loss of function. Moderate means that variant represents missense type of mutation. And modifier means that variant present in regulatory region. And low means that variant doesn't cause anything to harm. So we particularly focusing on high, moderate, and modifier. So the copy number variation analysis reveals the uh, number of copies of the genomic region is present. So we are deployed, the cattle also deployed organism. So which means the particular gene having the two copies. So if the region having more than two copies means we call it as copy number gain, and less than two copies means we call it as a copy number loss. So this analysis reveals Sahiwal having higher number of CPG, whereas Friesian having higher number of CPN, which indicating the lot of deletion which are covered post-mutation. So here I am showing the result of the reads mapped with the Darwin reference. So first we identify the genes having a variant in all three locations, and then we identify the common genes that are present in all three locations because this common thing confidently represent the particular gene. So the Delhi analysis revealed Kangayam and Tharparkar having higher number of duplication, whereas here having higher number of deletion. So this is the another gene list identified by type 3 base. So here we particularly focusing on single nucleotide polymorphism SNP. So we identify the common SNP that shared across all indigenous genes. So we found 1,600 genes that have a common SNP. From that 103 are immune genes containing 46 are innate immune genes. Then I mapped this 103 with Karan and Holstein and found 40 are innately present in indigenous breeds. So in that card, D9 and NLRP8 having a higher impact. So the FST analysis I did with gene level and also with sliding window analysis. So in that I have found PRKCA having a score of 0.1 and beta defensive having a score of 0.3. If the score more close to 1 means the gene has a higher genetic differentiation. So the beta defensive also follow the top 5 category. So I did the functional and pathway analysis of these 1,600 genes and found most of the genes fall under the biological process of regulating the immune process and in the pathway of JAK-STAT signaling pathway and cytokine receptor and others. So I also built a network for these 1600 genes having a common receptor and found three topological parameters like degree, bottleneck, and closeness. In all these three categories, Jack and TVK genes having a higher number of interaction which represent degree and having higher number of shortest path to other nodes act as a bridge which represent bottleneck and more closeness to other nodes. So in addition to that, I also found top three clusters in the network. So the top one cluster represents the JAXAT signaling. Overall, my analysis identify the genes having a significant genomic differences as well as their biological processes and pathways. And the network analysis revealed the immunogene as a hub in the network. So finally, I conclude my talk with acknowledgement. I have to thank ICAR NASF for funding, Sastra Deemed University for research support, and I welcome Jean ISCB for the opportunity. Thank you.